Today, we're going to take a guided tour inside the cell, looking at all the structures that make life possible at the most fundamental level. Cells may be microscopic, but they are very complex and highly organized. Each structure inside the cell has a specific role, and together they allow the cell to grow, respond to their environment, use energy, and reproduce. As we move through this topic, the goal is not to memorize parts, but to understand how the components work together as an integral system. By the end of this section, you should be able to connect all cell structures to cell function and appreciate how differences in the cell help explain the diversity of life the way we see it. When we zoom all the way out to the cellular level, we are really looking at the most basic units of life. Each living organism on Earth, from bacteria to humans, is made up of cells. Some organisms are single celled and carry out all of life's processes within one cell, while others are multicellular and depend on many specialized cells working together. The central idea in biology, known as cell theory, explains that all living things are composed of cells, and new cells arise from pre existing ones. Cells do not appear spontaneously. Every cell in your body today formed through the division of a previously existing cell. This means there is an unbroken chain of cell division connecting modern life to the earliest cells that ever existed. When we talk about cells, we're really talking about the world that exists far beyond what our eyes can see. As a general rule, objects about half a millimeter in size or larger are visible with the naked eye. Once we move beyond that threshold, we need microscopes. Objects between 1 millimeter and 100th nanometers can be seen using a light microscope, which is what we commonly use in the teaching and research labs. Anything smaller than that, down to about 1 nanometer, requires an electron microscope because light simply cannot resolve structures that small. To set a sense of scale, it helps to move from large to small. Humans are on the meter scale with an average height of about 2 meters. Some nerves and muscle cells can actually reach close to a meter in length. A chicken egg is several centimeters long, while frog eggs are only a few millimeters. Single cell organisms like paramecium fall in the 1 micrometer range, along with most plant and animal cells. As we move smaller, we find structures like the nuclei, mitochondria, and bacteria, followed by viruses, ribosomes, proteins, lipids, and finally the individual molecules of an atom. Biologists organize all living organisms into three major domains: bacteria, archaea, and eukarya. These domains reflect fundamental differences in cell structure. Organisms in the domain bacteria and archaea are made of prokaryotic cells, which lack membrane bound organelles in the nucleus. Their genetic material is located in the region of cell called the nucleoid. In contrast, organisms in the domain eukarya include protists, plants, fungi, and animals, are all composed of eukaryotic cells. These cells are larger and more complex, containing membrane-bound organelles such as the nucleus, mitochondria, and endoplasmic reticulum. This distinction between prokaryotic and eukaryotic cells is one of the most important organized principles in all of biology. A prokaryotic cell represents the simplest form of cellular life, but simple does not mean unsophisticated. These cells have a plasma membrane that controls what enters and leaves the cell, and many also have a rigid cell wall that provides protection and shape. Inside the cytoplasm contains ribosomes, which build proteins, and the DNA is concentrated in the nucleoid region rather than enclosed in the nucleus. Some prokaryotic cells have a structure like flagella for movement or pili for attachment. Despite their small size, prokaryotic cells are incredibly diverse and metabolically versatile, allowing them to thrive in nearly every environment on Earth, including in extreme conditions. Animal cells are eukaryotic cells that lack a cell wall and are built for flexibility and specialization. They contain a nucleus that houses DNA and coordinates cellular activity, along with the mitochondria that generate ATP to power cellular work. Other organelles, such as the endoplasmic reticulum and Golgi apparatus, handle protein and lipid production and distribution. Lysosomes are especially important in animal cells, breaking down waste materials and recycling cellular components. Because animal cells do not have a rigid cell wall, their shape can vary depending on their function, which is essential for processes like movement, communication, and forming complex tissues. Plant cells share many features with animal cells, but they also have a structure that reflects their unique lifestyle. A rigid cell wall made up of cellulose surrounds the plasma membrane, providing support and protection. Plant cells contain chloroplasts, which carry out photosynthesis and convert light energy into chemical energy stored in sugars. A large central vacuole that occupies most of the cell's volume, helping maintain internal pressures, stores material, and also regulates water balance. Together, these features allow plants to support upright growth, produce their own food, and survive in wide ranges of environments. Every living cell is separated from its surrounding by the boundary called the plasma membrane. This membrane is not just a passive barrier, it plays an active role in regulating what enters and leaves the cell. The membrane is primarily made up of phospholipids arranged in a double layer known as a phospholipid bilayer. Each phospholipid has a hydrophilic head that interacts with the water and the hydrophobic tails that avoid water, which causes them to naturally form this two-layer structure. Embedded within this bilayer are proteins that carry out specialized functions such as transport, signaling, and cell recognition. Because the components can move laterally, the membrane is described as a fluid mosaic, highlighting both its flexibility and its complex composition. The plasma membrane structure directly explains how it functions. The hydrophobic interior of the phospholipid bilayer acts as a selective barrier, preventing many polar or charged substances from crossing freely. At the same time, membrane proteins provide controlled pathways for specific molecules to move in or out of the cell. Some proteins function as channels or carriers, others act as receptors that receive signals, and some help anchor the cell to its surroundings. Cholesterol molecules, especially in animal cells, help maintain membrane fluidity under different temperature conditions. The outer surface of cells varies depending on the type of organism. Plant cells have a rigid cell wall made up of cellulose fibers that surround the plasma membrane. This cell wall provides protection, maintains cell shape, and prevents excessive water loss. Animals lack a cell wall, which allows for greater flexibility. Instead, many animals can secrete an extracellular matrix, a sticky network of proteins and carbohydrates that help cells attach to one another and communicate. Animal cells also form specialized connections called cell junctions, which physically link neighboring cells and allow tissues to function as coordinated units rather than an isolated cell. The nucleus functions as the control center of the eukaryotic cells because it houses the cell's genetic materials. DNA stores the instructions for building proteins, and proteins carry out most of the work inside the cell. The nucleus is enclosed by a double membrane called the nuclear envelope, which separates genetic material from the cytoplasm. Nuclear pores are embedded in the envelope, regulate the movement of molecules in and out of the nucleus. Ribosomes, which are responsible for protein synthesis, rely on genetic instructions that originate in the nucleus, linking DNA directly to cellular structures and functions. Inside the nucleus, DNA does not float freely. It is packaged with proteins into materials called chromatin. When a cell is not dividing, chromatin appears as long, thin fibers but during cell division, it condenses into a distinct chromosome. Each chromosome represents one long DNA molecule containing many genes. Also found within the nucleus is the nucleolus, a dense region where the components of the ribosomes are produced. These ribosomal subunits then exit the nucleus through the nuclear pores and assembles into the cytoplasm, where they begin to translate genetic information into proteins. Ribosomes are the structures responsible for building proteins, which are essential for nearly every cellular process. In eukaryotic cells, ribosomes components are assembled in the nucleolus and then transported into the cytoplasm. Some ribosomes float freely in the cytosol and produce proteins that function within the cell. Other ribosomes attach to the surface of the endoplasmic reticulum and produce proteins that are destined for the membrane, organelles, or secreted outside of the cell. Despite differences in the location, all ribosomes carry out the same basic task of translating genetic instructions into functional proteins. The information stored in DNA is not used directly to make proteins. Instead, DNA serves as a template to produce messenger RNA or mRNA in a process called transcription. This mRNA molecule carries a copy of the genetic instructions out of the nucleus and into the cytoplasm. Once in the cytoplasm, the mRNA binds to the ribosome, which reads the sequence of nucleotides and translates it into a specific sequence of amino acids. These amino acids are linked together to form a protein with a unique structure and function, connecting genetic information to observable traits. The endomembrane system is a network of interconnected membranes that work together to manufacture, modify, transport cellular products. This system includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosome, and the vacuoles. These components are either physically connected or communicate through small membrane-bound sacs called vesicles. By comparing and compartmentalizing different steps of production and transport, the endomembrane system increases efficiency and organization within the cell, allowing complex molecules to be processed and delivered to the correct destinations. The endoplasmic reticulum, or ER, is one of the cell's main manufacturing centers. It's a vast network of membranes connected together to the nuclear envelope and extending through the cytoplasm. The ER comes in two forms, rough and smooth, which differs in structure and function. Together, they produce a wide range of molecules, including proteins and lipids that are essential for the cell's structure and activity. The ER's close connection to the nucleus and Golgi apparatus allows materials to move efficiently through the cell's internal production line. The rough endoplasmic reticulum gets its name from the ribosomes attached to its outer surface. These ribosomes are actively synthesizing proteins that will either become part of the cellular membrane or be sent to other organelles, or to be exported out of the cell. As the proteins are made, they are instead inserted directly into the AR membrane or into the ER's internal space. Inside the rough ER, some proteins are chemically modified, folded into proper shapes, and prepared for transport. Once ready, they are packaged into transport vesicles that then bud off and carry these proteins to the next destination, usually the Golgi apparatus. Unlike the rough ER, the smooth endoplasmic reticulum does not have ribosomes attached to its surface. Its functions are very different and focus mainly on lipid production. The smooth endoplasmic reticulum synthesizes lipids such as phospholipids and steroids, which are essential components of the cell membrane and signaling molecules. In liver cells, the smooth ER plays a critical role in detoxifying drugs and harmful substances, making them easier to remove from the body. In some muscle cells, it also stores and releases calcium ions, which are important for muscle contractions. The Golgi apparatus acts as the cell's shipping and receiving center. It receives proteins and lipids from the ER in transport vesicles and then modifies, sorts, and packages them to their final destination. As molecules pass through the Golgi, they may be chemically altered, labeled with molecular tags, or assembled into more complex structures. Once processing is complete, the Golgi directs these products to a specific location within the cell or packages them into the vesicles for secretion out of the cell. This precise sorting system ensures that each product ends up where it needs to go. Lysosomes are membrane-bound sacs filled with digestive enzymes and are primarily found in animal cells. These enzymes are capable of breaking down large biomolecules such as proteins, lipids, and carbohydrates and nucleic acids. Lysosomes function as the cell's recycling center, breaking down worn-out organelles and cellular debris so their components can be reused. They also help destroy harmful bacteria and other pathogens that enter the cell, playing an important role in cellular defense and maintenance. Beyond digestion and recycling, lysosomes are also essential for normal development and health. During embryonic development, lysosomes help sculpt tissues by breaking down specific cells, such as those between the developing fingers. Problems with lysosomal functions can lead to serious genetic conditions known as lysosomal storage diseases. In these disorders, materials that should be broken down accumulate inside the cell, often causing severe damage to the tissues and organs and highlighting how critical lysosomes are for normal cellular function. Vacuoles are large membrane-bound compartments that serve different purposes depending on the type of cell. In many single-cell organisms, food vacuoles form when the cell engulfs nutrients, allowing digestion to occur internally. Some freshwater protists have contractile vacuoles that actively pump excess water out of the cell, preventing it from bursting due to the osmotic pressures. In plant cells, the central vacuole is especially prominent that can take up most of the cell's volume. This structure stores water, nutrients, pigments, and sometimes toxic components that deter herbivores. By maintaining internal pressures, the central vacuole also helps keep plant cells rigid and supports overall plant structure. When we step back and look at the endomembrane system as a whole, it functions like a coordinated assembly line. The nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles work together to manufacture, modify, transport, and recycle cellular material. Proteins and lipids are synthesized in the ER, processed and sorted in the Golgi, delivered to their destination by vesicles, and eventually broken down, recycled by lysosomes when they are no longer needed. The central theme in biology is energy transformation, and cells rely on specialized organelles to manage this process. Chloroplasts and mitochondria act as the cell's power centers, converting energy into usable forms. These organelles allow them to capture energy from the environment or extract energy from their molecules. Without them, complex life as we know it would not be possible. Their presence highlights how cells are not just structural units, but the dynamic systems constantly processing energy to sustain life. Chloroplasts are found in the photosynthetic cells of plants and algae and are responsible for photosynthesis. During this process, light energy from the sun is converted into chemical energy stored in the sugars and other organic molecules. These sugars serve as the foundation for most food webs on Earth, directly or indirectly supporting nearly all living organisms. Chloroplasts contain internal membranes that minimize the surface area available for capturing light, making photosynthesis highly efficient and central to life on this planet. Mitochondria are present in nearly all eukaryotic cells and are the sites of cellular respiration. Here, energy stored in food molecules is converted into ATP, the molecule cells use as their immediate source of energy. ATP powers the processes such as muscular contractions, active transport, and biosynthesis. Cells that require large amounts of energy, like muscles or nerve cells, often contain many mitochondria. Chloroplasts and mitochondria share several unusual features that set them apart from other organelles in the cell. Both contain their own DNA and ribosomes, which allow them to produce some of their own proteins independently of the nucleus. The DNA found in these organelles is circular and resembles DNA of prokaryotic cells, rather than the linear chromosomes found in the nucleus. In addition, chloroplasts and mitochondria can grow and divide on their own within the cell. These similarities suggest that they have a different evolutionary history compared to other organelles. The unique features of chloroplasts and mitochondria provide strong evidence for the endosymbiotic theory. This theory proposes that these organelles originated as free living prokaryotic cells that were engulfed by a larger host cell. Instead of being digested, they formed a mutually beneficial relationship within the cell. Over time, the relationship became permanent, with the host cell and the internalized cells becoming increasingly interdependent. Eventually, this partnership gave rise to modern eukaryotic cells with chloroplasts and mitochondria as integral components. The cytoskeleton is a dynamic network of protein fibers that extend throughout the cytoplasm of the cell. It functions both as supportive framework and as a system that enables movement. By providing structural support, the cytoskeleton helps the cell maintain its shape and internal organization. At the same time, it also allows the cell to move, change shape, and transports materials within the cytoplasm. Rather than by being rigid, the cytoskeleton is constantly being assembled and disassembled, allowing cells to adapt to their environment. The cytoskeleton is made up of several types of protein fibers, each with specific properties and functions. Microtubules are hollow tubes that help resist compression and serve as a track for motor proteins that move organelles around the cell. Intermediate filaments provide tensile strength, helping cells withstand the mechanical stress. Microfilaments, which are thinner, play key roles in cell movement and cell shape. Together, these fibers anchor organelles in place and coordinate internal organization, ensuring that the cells function efficiently. Some eukaryotic cells have specialized structures made up of microtubules called cilia and flagella. These structures extend from the cell surface and are involved in movement. Flagella are typically long and few in number and propel cells through a whip-like motion as seen in the human sperm cell. Cilia are shorter and more numerous and move in coordinated waves. Both structures allow cells to interact with their environment, either by moving the entire cell or moving fluid around the cell surface. Cilia are short hair-like extensions that project from the surface of the most eukaryotic cells and play an important role in movement and fluid transport. Unlike flagella, which usually move a cell through its environment, cilia often move substances along the surface of the cell. They beat in a coordinated back and forth pattern similar to a synchronized motion of oars on a rowing team. This coordinated movement allows cilia to efficiently move fluid or particles in a specific direction. Cilia are also found in many single-celled organisms, where they can help propel the organism through water and assisting in feeding. Cilia and flagella are not just important for movement, but also for normal human health. In the respiratory tract, cilia line the airways and continuously sweep mucus trapped particles out of the lungs, helping keep the airways clear. When the structure and function of cilia or flagella is defective, serious health problems can occur. For example, problems with flagella can lead to male infertility because sperm rely on flagella for movement. Some individuals also experience chronic respiratory infections due to impaired cilia function, illustrating how essential these structures are beyond simple cell motility.